Please give a warm welcome to HDSA's Chief Mission Officer, Dr. Eric Johnson.
Thank you. I love that. It feels like I know sports or something, but yeah, go sports, balls. This is awesome. Hey, everyone. Good morning, and welcome to day 2 of the 37th Annual HDSA Convention. I am Dr. Eric Johnson, and I'm the Chief Mission Officer at HDSA. It is my pleasure to serve the HD community by overseeing the mission team and all of the HDSA's programs and services. These cover things in the following areas: education, clinical care, social services, disability, advocacy, youth services, and research. It's a mouthful. You trip over it every time. I hope you take the opportunity to meet the members of the HDSA mission team this weekend and all of the HDSA staff who's here. And you can always find out more about what we do on the HDSA website, www.hdsa.org. Before I say too much more, I'd like to remind you about a couple of things this morning. First, I want to thank you all and remind you all to continue wearing your mask whenever you're in an HDSA convention activity, unless you're eating or drinking. Next, for questions you have for this upcoming talk or in any of the sessions that you participate in, please make sure to do so through the HDSA Convention Program app. How you do that is you open the app, click on Agenda, and then click, for this case, on the Research Keynote. You'll then see Q&A right at the top of the page, and when you click on that, you're able to type in your questions, which we'll sort through and we'll ask. I hope you were all able to attend the NYA talent show last night. It was so great to see the NYA back in action. It was great to see some pretty incredible performances. And more important than anything, it's an amazing event to raise funds for the National Youth Alliance, specifically for scholarships to convention. So last night, they raised over $21,000 to support convention scholarships for next year. Thanks to everyone who attended, whether you were here in the room or watching at home. Thank you to everyone who donated. Continue to support that group. They are amazing. Help support them in providing a community and family for young people who are living with HD. Now, I was going to say I'm excited to tell you a little bit more. I also want to tell you it's awesome to be back in front of people like this. It's not awesome to be in front of you because that's really nerve-wracking. It's awesome to be here with you all. I want to talk a little bit about the HDSA research programs today. Our commitment to HD science and research can be seen in a number of different ways. First, HDSA directly supports research through 3 different grant programs. Each of these programs fills a critical gap in the HD science landscape by attracting young scientists to our field and answering key questions in human biology that are critical to developing disease-modifying treatments. The Donald A. King Summer Research Fellowship is a 10-week grant for college students working under the supervision of senior HD scientists on a focused project that, while it's conducted in a short period of time, will be helpful in guiding future HD science. This fellowship aims to attract the brightest young scientists to Huntington's disease research, many of whom stay on in the field because of the experience that was made possible by this HDSA grant. Next is the Berman Topper HD Career Development Fellowship. HDSA believes we must ensure that the pipeline of passionate and bright HD scientists and clinicians remains full, a vision shared by the Berman and Topper families. Together, we're committed to championing young research with a desire to make HD part of their career plan. And the goal of this fellowship is to develop and support the next generation of passionate and innovative HD scientists. This is an awesome group of people, y'all. So the Berman Topper Fellows have become an amazing brain trust who collaborate together as a group of scientists to change the future of HD science. Last and certainly not least is the HD Human Biology Project. HDSA launched this program back in 2013, which promotes a patient-centric research strategy to push the field closer to meeting our goal of identifying effective therapies. The Human Biology Project supports researchers working to better understand the biology of Huntington's disease as it occurs in human beings, and who are doing that work in collaboration with established HD clinics, either within the HDSA Centers of Excellence network or at HD clinics around the world. Over the years, research funded by this program has been fundamental in the identification of new biomarkers for HD and for potential therapies. Recipients of the HDSA grant awards have gone on to successful careers and are emerging leaders in the HD science field. If you attended any of the research sessions yesterday or do so today, you're actually going to have a chance to meet some of them right here in Atlanta. In addition to directly supporting research through these grants programs, HDSA knows that HD families are critical partners in HD science. Without brave people like yourselves, those of you here in the room, those of you watching from home, without you choosing to participate in clinical research studies, new drugs to treat HD, and hopefully one day stop it, can't be developed. We work hard to help you understand the science, answer your questions, and empower you so that you can be excited about all of the research that's going on. And if you're interested and able to do so, get involved in a clinical study. One way HDSA shares research information with the community is through scientific communication. From the HDSA This Week in HD research blog, to webinars featuring breaking news, trial updates, and more, to our partnership with HD Buzz, we want to make sure people living with HD know what's happening in HD research when it's happening. You're also going to see this another way today when Dr. Leora Fox will be hosting the Clinical Trials Showcase and Luncheon. This is an opportunity to learn directly from some of our pharmaceutical companies working in the HD field about their currently enrolling clinical trials. Whether you're considering joining one of these trials, if you've already participated, in the CHDI blood draw, or you're going to do it when you have a break today, it's going to be open until 3 o'clock, by the way, so please, please, please go. Or if you are a part of the many studies that are taking place at our centers of excellence across the country, your involvement in HD research is greatly appreciated. We want to support and empower you to take part in clinical trials. Like I said, without your participation, many of the studies and trials could not happen. They wouldn't happen if you weren't there. You are the key to finding better treatments and eventually curing HD. This brings us to this morning's research keynote speaker, and an opportunity to learn about something that will help direct HD science in clinical trials as we pursue new and better ways to treat Huntington's disease, both symptoms and eventually the disease itself. While we often focus on the drug trials that bring treatments closer to use in clinics, there's a lot of work that has to be done to get to that point. Whether that's ongoing exploration of how Huntington's disease works on a cellular level, learning more about disease progression through computational modeling, or conducting translational research that moves science from the bench in animal models into humans. This work strengthens the foundation of HD science to bring us closer to treatments and a cure. The HD Integrated Staging System, the HDISS, is an example of something that will impact clinical research, both in how studies are designed and how treatments are developed. You got a bit of a sneak peek about this yesterday morning at the opening ceremony, but now you're going to hear about it in more detail. The HDISS is a critical tool for HD research moving forward as a way of defining distinct study populations across the entire disease progression, including people who might not have motor symptoms yet or a clinical diagnosis of Huntington's disease. Here to tell us more about the HDISS is Dr. Sarah Tabrizi. Dr. Tabrizi is the director of University College London Huntington's Disease Center, joint head of the Department of Neurodegenerative Disease at UCL Queen Square Institute of Neurology, a principal investigator at the UK Dementia Research Institute, an honorary consultant neurologist at the National Hospital for Neurology and Neurosurgery. She's kind of a big deal. Dr. Tabrizi started her studies in biochemistry and then medicine at the University of Edinburgh, where she was recognized the most distinguished medical graduate. She undertook her PhD and trained as an independent scientist at UCL, where she focused on finding disease-modifying therapies for HD. Dr. Tabrizi was PI on the first successful phase 1/2b trial of an antisense oligonucleotide and several other pivotal clinical studies. She also currently serves on a number of scientific advisory boards, advising industry on the development of potential gene targeting and nucleic acid therapies for HD. Dr. Tabrizi has published over 350 peer-reviewed articles and research papers, is a fellow of the UK Academy of Medical Sciences, and has received numerous awards from organizations including the Hereditary Disease Foundation, the Spanish Society of Neurology, the Royal College of Physicians in Edinburgh, the Association of Physicians of Great Britain and Ireland, and the World Congress of Neurology. I said before, kind of a big deal. She's a big deal, you guys, and we are so lucky to have her here today joining us. We're honored to give her a warm welcome to the HDSA National Convention. Welcome, Dr. Tabrizi. Thank you so much, Sarah.
Thank you. Thank you. Thank you so much. And thank you, Eric, for such a lovely introduction. And I'm very grateful. I'm really excited to be here. I was so happy when I got Louise and Eric's invitation. And I'm excited to tell you about the HDISS, or the International Space Station, if you want to. Okay, so you've heard about it a lot. So what I'm going to do over the next kind of 35 minutes is deep dive into the nitty-gritty. And at the end, there is going to be loads of opportunity to ask any question that you want. So I'm going to talk about what it is, why is it important, what are its implications for persons with Huntington's disease. And I've put the talk together in a series of questions, which are questions that many people ask. So this is what I'm going to go through. What is clinical research? What is a medical staging system? And why is a new system needed? What about what we already have? So the HDISS, who created it? What is it? And how does it work? How can I be staged? Do I need an MRI? And does this change what we know about Huntington's disease? And so these are the questions that I'm going to go through, but you will probably have many more, and we have lots of time to answer them. So what is clinical research? So clinical research is basically research in human beings. It's used to advance medical knowledge, to do clinical trials, to test new therapies, to try and understand more about biomarkers. For a successful study in humans, you need to accurately select the correct participants that you want in your study. And you must be able to distinguish healthy people from people with a medical condition to be able to do any form of clinical research. So that's the kind of fundamentals. So what is a medical staging system? And I'm going to give you some examples so you can understand how it's used and has revolutionized a number of areas in medicine. So a medical staging system is a staging system that divides human beings into groups with similar characteristics. And you can see here, this is a group, and these are just different stages of that group depending on similar characteristics, a very simple example. So each group or stage has similar characteristics, common problems, and may benefit from similar solutions and treatments. And you need to understand, so to be able to do this staging, you need to have a deep dive into understanding which characteristics best define those groups. So what is a medical staging system? So I think in terms of myself and probably many people in this room, many people are touched in their lives by people, someone they love or someone they know who has cancer. Cancer, the medical staging system of cancer has revolutionized treatment of cancer. It's a universal staging system, it's used all over the world, and it's directly comparable. And I'm going to talk through that so you understand why we need a staging system. And so stage 0 cancer means a very small cancer that's found locally. It's all dependent on the size. Then stage 1 is it's slightly larger. Stage 2, it might have spread to lymph nodes. Then stage 3, it can go a little bit wider spread in terms of size. And then stage 4 is when it's spread throughout the body. That is used for every cancer, regardless of where it comes from, in every human being and in every study. And it has truly revolutionized the treatment of cancer. We know that breast cancer is treatable because we know the treatments for each of those stages. So this is really an important example, but an example that has a massive impact on the field. So as I said, stages divide people into groups with similar characteristics. Each group may benefit from specific treatments, as in the difference between treatment of a stage 0 breast cancer and a stage 3. And you need to understand what defines those groups. In cancer, it's size and spread. And the FDA, and I'm mentioning the FDA because this is an American audience, the regulators want clear and accepted staging systems that cover the totality of a disease. And we know now in cancer that there are huge cancer screening programs where people are being screened for the possibility of having cancer so they can have preventative treatment. And that has all been set up because we have staging systems and the regulators understand the importance of it. So in Huntington's, let's bring it back to Huntington's disease. Why do we need new? What about what we already have? Before the discovery of the HD gene in '93, there were decades of research on human beings. But researchers still had to be certain for their work who did and who didn't have the gene, because there wasn't a test. And that resulted in diagnosis, or DCL4, which some of you will have heard of, which is the diagnostic confidence level, and it is made when HD symptoms are absolutely unmistakable. And typically, that's more in the whole lifespan of an individual, that's more in the more advanced stage of the disease. And it led to a clinical diagnosis that we all know based on motor features that are unmistakable. So why don't we just use that? So what about clinical motor diagnosis? Relying on this for research is difficult. It's very helpful in the clinic when you're seeing patients, and we see patients and we do the total motor score and the UHDRS every time we see them. But relying on this for research is difficult for a number of reasons. The diagnostic criteria for that are subjective. So if I examine someone, do their motor score, and then look at the DCL, it goes from slight thoughts of the patient having Huntington's to 50% likelihood, 75% likelihood, greater than 90% likelihood. And so it's very subjective. So if I do that score with an individual, it might be different from the score that Chung Ho does. Or it might be different from the score Ed does. And that is what we mean by subjective. It's also dependent on the person doing it. And so it's not an exact science. And we all use the terms, and Ed mentioned this in the HD Buzz presentation yesterday. We use onset, we use pre-manifest, and I have used all of these terms in my research. We use prodromal, and we use manifest, symptomatic, pre-symptomatic. They're useful in the clinic when you're seeing patients to try to help explain things, but they don't have precise definitions. What do we really mean by prodromal? We don't have an exact definition of it. What do we mean by manifest? Is it motor manifest, or is it when there may be some thinking problems? And so they're very woolly definitions, which isn't good for science. And comparing results across studies is quite challenging because you don't know exactly what each study uses to define the criteria. So you can't compare them head to head. And we know that the motor diagnosis occurs relatively late in the entire lifespan of an individual with Huntington's disease. We know, and this is something that patients and families have been telling us. I started my PhD in 1996, and I started working with patients and families then, and it was very clear, and we listen and we know that signs and symptoms, for example, thinking problems, personality problems, maybe personality changes, maybe not being able to multitask so well, not coping with stress so well. They're very subtle, but many people complain of some thinking and personality changes and earlier than when they have any motor signs. And we also know we know a lot about the biology of Huntington's disease from cells and mice and you name it. We have animal models of every system. We have pigs and monkeys and mice. But for me, it's the human being that is really the most important because they're the people who have Huntington's disease. You know, mice don't get Huntington's disease. They are a model. And so we know that underlying biological changes exist much earlier in human beings. And there's a huge amount of work that's been done on that. Currently, and Eric mentioned this and Ed mentioned it, it is very difficult for trials to include earlier participants. We don't have the regulatory framework for it. And we want to do earlier stage clinical trials as this really may be the best chance for us to make the biggest impact on slowing or stopping this disease. And after being in this field now for 26 years, I feel very passionately about this. So again, why is new needed? What about what we have? So many of you will have heard of things like the CAP score and the PIN score. And the CAP score has commonly been used to recruit for clinical trials. So what is it? So these are just mathematical equations. They're pretty difficult to understand. They come from mathematical models. And the CAP uses the age and the CAG size, and the PIN uses the age, the CAG size, and clinical scores. They're difficult to understand. If you talk to someone outside the HD field, or you talk to regulators, or you talk to people who don't know anything about Huntington's disease, they have no idea what you mean when you say CAP and PIN. So we need to have better language. And it may not be sufficient for selecting participants accurately. They can be useful. I think the PIN is going to be particularly useful to stratify and enrich groups, and I'm happy to talk about that. But we just don't think they're useful for staging at this stage. They do have uses, but not for staging. So why don't we just develop a very specific definition for our own particular study? Why don't we just say, this is who we want, and this is who we're going to define? And there's a big problem with that, because if everyone did it, we would never be able to compare trials or studies, and you just couldn't compare data. You couldn't do what's called a meta-analysis, where you put all the data together and look at what it means in large numbers. And it may not be sufficient for selecting participants accurately. So I hope I've managed to convince you that we do need new. So who created the HDISS? So this work started 3 years ago. It was done with the Huntington's Disease Regulatory Science Consortium, which was as part of the Critical Path Institute, which has close links with the regulators, the FDA in the US and the European Medicines Agency in Europe. We had a critical path working group. I was the chair, and it was made up of HD researchers, clinicians, scientists, and representatives from industry who all had clinical programs in Huntington's disease, and it was a very good working group. It examined all the scientific evidence. It examined thousands of data sets to reach consensus conclusions. It had to be very objective, and we had to use very rigorous scientific methods. And we also had critical input from HD patients and families. We had meetings with HD COPE. We presented it to HDSA, European Huntington's Association, HDYO, and the Huntington Society of Canada, and also the regulators at the FDA. And it's going to be published on the 15th of June, next Wednesday, so all of you will be able to get a copy. So how did we learn all this about Huntington's disease? And this is where all of your participation and the participation of every person throughout the world, patient, family, a family member, friend, who has given their time, days or 2 days, to contribute to HD research. All this is possible because of your research participation and the participation of patients and families globally. Enroll HD, we used all that data, 20,000 Enroll HD participants. We used all the data from Image HD, Predict HD, many of you will have been in the critically important Predict HD study. Track HD, that was another very important study. We used all that data. And Cohort, which many of you were involved in. We used all that data, and I want to thank you for participating in that clinical research, giving up your time, which allowed us to do this. So thank you. This is really important. So what is it? It combines what we know about the genetics. It combines what we know about biomarkers, and it combines what we know about clinical signs and symptoms. And I'm going to go through this bit by bit. It defines Huntington's disease on the basis of the genetic test. The HDISS divides Huntington's disease into 4 stages. Stage 0, 1, 2, and 3. And it covers the full lifespan of a person with Huntington's disease. And the people in each stage share predefined common characteristics. So why is it important for clinical research? It's important for easy communication with regulators, across trials, with industry. One day when we have a potential disease-modifying trial, it's going to be important for talking to people who are going to pay for the medicines. And so it's critical for our communication. The HDISS was developed to provide a universal tool for research in Huntington's disease, like the staging system that covers all of cancer. We have an unambiguous definition of who is a person with HD. It is standard across all geography, all studies, and is a very clear communication. Remember, this is research. This is for clinical research. It allows now for the entire lifespan from birth of a person with HD to be included in trials. Now, clearly, we aren't going to be doing in trials in very young children who are at stage 0 at this stage, but it allows us to have the framework to do trials in young people with Huntington's disease who may have juvenile Huntington's disease, for example. And I know many of you are in the audience. It enables clinical research in early HD clinical phases, and I think that's critical. So how does it work? Huntington's disease is defined as the presence of a CAG repeat expansion of 40 or more in the Huntington gene. It's a biological definition. We know that there are some people who have between 36 and 39 CAG repeats. We know that some of those people won't go on to develop Huntington's disease in terms of the progressive features of Huntington's disease. And that's because it's called what's called reduced penetrance. So they're only included in the definition of HD if they have something extra. They have to have additional signs of biomarkers or clinical signs and symptoms that are consistent with the diagnosis of Huntington's disease. So it's a separate group. So this is a map of the HD integrated staging system. And I'm just going to talk you through it, but I'm going to talk through how we came to this. So it's quite simple, because staging systems are best simple, like the cancer staging system. And we know that there is a trajectory or a lifespan of a person with Huntington's disease. And we know that it follows a progressive course. So this is stage 0, the Huntington gene, nothing else. And actually, we had a lot of discussion with patients and families because this was originally called stage 1. And myself and many people in the patients and families preferred the stage 0. Absolutely no biomarkers of pathogenesis at this stage could be detected based on the available data we have to date. Then there's stage 1, then stage 2, then stage 3. Stage 1 is when a biomarker of pathogenesis is detectable. Stage 2 is when there are clinical signs and symptoms detectable. And stage 3 is when there is some impairment of function or some loss of independence. We know from decades, actually centuries, of what we know about patients with families with Huntington's disease. Even George Huntington wrote about this in 1872, that it follows this. It follows this course. Sorry, it's not. So we know that it follows this course. So these are the gates, and there are 2 gates at each stage, and I'm going to come on to talk what the gates are. The gates are basically landmarks. But we know in the course of the disease that some people don't go just straight like that. That's quite uncommon. Most people go on a path. And some people can go more quickly to stage 1, and some people can go more slowly to stage 1, and so on. And so the path for each person is different, but that it does always go in this progressive order. And we know that each of these stages predict the next stage. If you have the HD gene at stage 0, we know from thousands of data sets that there will be this biomarker and that there will be signs and symptoms. So they have predictive value. We know that they differ in the path, and that's by many things. It can be genetic modifiers. You've all heard a lot about genetic modifiers. They are very big news. It could be environment. There's many things that can affect how quickly or slowly someone progresses. So let's come back to the landmarks. Think of the landmarks as gates or flags. So stage 1, biomarker of pathogenesis. Stage 2, clinical sign or symptom. Stage 3, functional change. That's very simply in a nutshell. So this is how we did it. We did a literature review of every potential assessment that had ever been done in thousands of human beings with Huntington's disease, and families with controls, family members without the Huntington gene, but also many, many spouse, partners, friends who were in clinical research. It was completely data-driven and objective. We didn't just choose things. And we had 2,790 landmarks from data that we had. We then decided, because that was too many, and we didn't want this complicated, it's like cancer staging is just size, we chose ones that had over 100 participants and longitudinal data, so had been assessed over time. And that gave us 371. There was 176 that were unique, and 27 that were predictive, which prognostic. For example, brain imaging is predictive. So that is a very important biomarker. And there were 16 that were strong. So we narrowed the criteria, and we also wanted criteria that had been studied in more than 2 studies, ideally, or in one model that had more than 1 data set. So it was quite complicated, but we had to be really, really, really rigorous. We couldn't just sit around a table and think, this is what I know about seeing patients and families with Huntington's disease. That was not how it was done. It was a really, it was a proper scientific experiment. I run a lab, we had a hypothesis, we tested it. And the landmarks were then nominated. We had 16, and we voted on by the group through an NIH consensus process. So it was completely objective. So, stage 0, the presence of the gene of CAG more than 40. No detectable biomarker of pathogenesis. Biomarker. The landmarks were caudate volume and putamen volume, and this was from thousands of scans. And the best biomarker, actually, that we have currently that is very predictive of disease is brain imaging. Now, many of you asked me about CSF neurofilament or plasma neurofilament. I think it's very important. We just don't have the samples of thousands of samples longitudinally in more than 1 data set. So that will come, and it may be that we use, and I'm a big fan, maybe it will be that we use markers like CSF neurofilament to actually tell us more about subgroups in stage 0 and possibly also in stage 1. And I think that's what will happen. But we don't have the data for it yet, and it has to be very rigorous data with very validated methods of quantification. Again, CSF mutant Huntington. CSF mutant Huntington is another marker, but we still don't know enough about it, where it comes from, how it tracks with disease, and there needs to be more work in larger numbers in CSF over longer periods of time to understand more about it, and indeed if it's associated with clinical progression or is prognostic. So the biomarker that really stood up to very high rigor was brain imaging. And those landmarks are caudate volume and putamen volume. And then clinical signs and symptoms. There were literally thousands of clinical signs and symptoms that had been tested in lots of studies. Tapping, lots of cognitive tests. And the 2 that really stood up to very high rigor was our good friends Total Motor Score and Symbol Digit Modality Test. And both of them are very powerful tools in that they were very predictive of future progression, but also very clear that they were the landmarks. So the landmarks for clinical sign and symptom is total motor score and symbol digit modality test, which is a cognitive test, which you do often on a laptop, but often with a pencil and paper. And I'll tell you a bit about how that was done. And then functional decline, which is stage 3, which is any change in the TFC, 13 to 12, which is a big, important change, or any drop in independence from below on the independence scale, so below 100. So how do we determine cutoffs? Because we have to determine cutoffs to have these landmarks. And as I mentioned, I come back to the cancer staging, but I think it helps to understand their cutoffs were size and spread. So we had to determine cutoffs. So luckily, because many people who participate in clinical trials are family members who don't carry the Huntington's disease gene, friends, spouses, partners, all the people who have a CAG less than 36. And their participation was really important. So what this let us do was model all of these measures, because we had data on all of these people. And we did it a bit like the WHO growth standards. So all of you who have kids will know when you go to the pediatrician or the district nurse, they weigh your child. And so this is the WHO growth curve. For a 10-month-old baby or child, if they weigh 8.5 kilograms, they're here on the blue line, which is the 50th percentile, which is average. All good. If you have a 10-month-old child, but actually they weigh 7 kilograms, for whatever reason, they're being sick a lot, that's actually the 5th percentile. And what we know is below the 5th percentile begins to become atypical. And so this is how we did the modeling. We picked a cutoff for what we described as an outlier, and we used the similar to the WHO curves. And if you scored beyond that 5th percentile, above or below the 5th percentile, dependent on the measure, for an assessment, it's not typical. And so this is an example relating to Huntington's disease. CAG 40. Caudate volume is below the 5th percentile for that age. And we have mapped these cutoff curves for everything. For caudate volume, with thousands of individuals who don't carry the Huntington's disease gene. Putamen volume, symbol digit modality test, total motor score, and we have the cutoffs. And those cutoffs then are calculated, and then if you're below the 5th percentile, that's the landmark. So this is all in one slide, and there's lots of figures in the paper if you want to refer to it. So we have stage 0, gene, no biomarker of pathogenesis. Stage 1, a landmark of below the 5th percentile of putamen and caudate volume. Now that's pretty subtle, but it's important and it's predictive. Stage 2, below the 5th percentile cutoff for any change in the total motor score and the symbol digit. And the cutoffs, as I say, have come from lots of normative data. And the landmarks, change in total functional capacity and independence scale. They are good landmarks for any change in function. And they're validated. And stage 3 divides into mild, moderate, and severe. So the questions. What is the impact of the HDISS for a person with HD? So this is part 1. How do I know my HDISS stage? Will I learn it if I don't want to? So, person with Huntington's disease. If you do not want to participate in any clinical research, you don't need the HDISS. You can forget about it. It has no impact on your routine clinical care. This is, which I showed you on my first slide, for clinical research. Yes, I do want to participate in clinical research. I want to be in an observational study. I want to give blood straight after this talk. I want to be in a trial. So you can either, and the HDSA and other organizations have find clinical trials links. You can shop for a study, a clinical trial or observational study. And the HDISS stage will be helpful to look for a good fit if you want to participate in clinical research, because the trials will say, we are recruiting HDISS of this stage. So it allows a comparator across everything, because it allows you to shop potentially for a clinical trial. If you want, you can ask for your HDISS stage from your doctor, because your doctor will know, because they will be evaluating you as part of Enroll and or evaluating you for clinical trials. To join a study that your doctor offers to you, your doctor will calculate your HDISS stage. If you want, you can ask your doctor, what's my stage? You may not want to know your stage, even though you want to be in the clinical trial. That's okay. But I just want to say you may figure out your stage if you look up and learn about what the criteria are to be in a trial. So you kind of may figure it out. So, but you don't need to know it. But as I say, you may figure it out. So what's the impact for me? Person with Huntington's disease. Do not want to participate in clinical research. You don't need to know anything about the HDISS stage, and it has no impact on your routine clinical care. You do want to participate. Your doctor can tell you your stage at your request and with your consent. So that's important. You have a choice. So many people have asked me, do I need to get an MRI? Because the MRI is done in a very special way, where the parts of the brain are measured. It's not like a routine clinical care MRI. Do I need to get an MRI to learn what stage I'm in? MRI is usually not needed at this stage. We can estimate HDISS stages using statistics. There are models that allow us to do that. And we can use clinical measures to deduce what stage someone is in, like the TMS or the SDMT cutoffs, because the stages go in order. So we can deduce, we can do the TMS and SDMT cutoffs for stage 2, and actually it might tell us the person is in stage 1, and they didn't need to have an MRI. The MRI is particularly important when it comes to stage 0 to stage 1. So these inexact methods are generally okay for just right now, because all current clinical trials are currently in HDISS stage 3 or HDISS stage 2, and the PTC study is going to be the first study to use the new HDISS staging system, and they are using the HDISS stage 2. More and more studies will do this. So we're ready for clinical trials now in HDISS stage 2, which is great. We are doing the work to do them really early in stage 0 and 1 to get the markers we need to be able to do clinical trials in that group. But clinical trials in those groups will happen, but at this stage in time, there aren't any currently planned, but there will be soon. So sometimes an MRI will be needed for confirmation and to learn precisely about a person's actual stage. And often now, when you are recruited for a clinical study, you will have an MRI as part of your screening. So, what is the impact? Does it change what we know about Huntington's disease? This doesn't change what we know about Huntington's disease. Historically, Huntington's disease was perceived as a condition that appeared decades after birth, but we know clinical signs and symptoms occur prior to clinical motor diagnosis, and I'm sure every person in this room who knows someone or is a person with Huntington's disease knows that. We know from a huge amount of biology, both in humans and model systems, that the clinical symptoms in humans are a consequences of changes in the brain structure. We know that. We have a lot of data to show that, which themselves are a consequence in the change in function of cells. So it's a whole biological continuum. And actually, the HDSA encompasses that biology. It incorporates and categorizes the underlying biology that allows us to do clinical trials much earlier in the disease process. So, in summary, it was developed, the HDSA was developed using evidence-based, state-of-the-art scientific techniques. It allows for accurate determination of disease status, and it allows us to define groups of persons with Huntington's disease with similar prognostic characteristics in those stages. It facilitates communication. We compare, for example, when we have the data, we compare 10 trials in HDSA stage 2. We will be able to directly compare them head to head. It facilitates communication with regulators, allows accurate selection of research participants. There is no gray zone here. The landmarks and the cutoffs are very clearly defined with cutoffs. And it allows us to compare across different studies. That's important. It's important for science. It's important for validation. It allows us to do clinical trials much earlier in the disease course. And we expect this to be one important means of getting the regulators on board with clinical trials much earlier in the disease course. In stage 0, in stage 1, and in stage 2, which is happening now, which is already much earlier in the disease course. And just to reiterate, and this is why your participation in research is so important, because that's how we move forward. We can't move forward as scientists and doctors without your help. This work would not have been possible without the vital contribution of the HD research participants and families. And the HD community's dedication to research has allowed the development of the HDSA. So I'll finish. I like to think of the world as one big laboratory. And I'm using this in the space theme that was started by Ed and Jeff yesterday. Our global community of HD families, scientists and clinicians working together is our chance of achieving success. And I think that time is now. Thank you. Thank you. Thank you. So I'd like to thank people, because this isn't a one-man show. I'd like to particularly thank 3 ladies who I have worked very closely with, Emily Gantman, Alexandra Mansbach, and Christina Sampao. And actually, Christina really helped spearhead this with her background in regulatory science and her experience of having done the staging system in Alzheimer's disease. And so we had a guiding influence with someone who knew what was needed for a staging system and with regulators. So I'm incredibly grateful to Christina in particular. So please have a round of applause. I'd also like to thank the other people who are part of the team. As I say, Emily and Lexi were amazing. Jeff Long did all those stats. Jeff Long did all the stats and was very patient. All of the other members, Scott Schobel, Beth Borowski, Pavlina, Tiago Mestre, Jennifer Panagoulis, Chris Ross, Morris Zoderer, Ariana Mullen, Klaus Romero, and Sudhir Sivakumaran and Emily Turner were all part of the working group, came from industry, came from academia, were doctors, were working in Huntington's research. We also had the input of the entire CPATH consortium and also, as I mentioned, the patients and families and impact from the feedback from the regulators. I'd also like to thank CHDI who funded the whole thing. So we have a big thank you to them. Without CHDI, we couldn't have done this. So I think we owe a huge debt. And in particular, Roby, Darren, Simon, Chris, and Swati. And again, we couldn't have done this without you. And so I'm happy to say thank you for listening, happy to answer anything you want.
Sarah, thank you so much. And now you just get to listen to the disembodied voice in the back of the room. So,
Oh, yeah, where are you? Okay.
I want to thank everyone who submitted questions, and there are some fantastic things that have been put into the chat. So I'm going to read some of those off to you. We've got about 15 to 20 minutes to answer those. So a couple of things have come in asking about people who are at risk or not tested, and really just what it means once you've gone through that process. So someone asked, it doesn't appear that this accounts for people who are at risk and not tested. What does that mean for their access to participating in research?
It's a good question. So currently, to get into any clinical trial, you have to have a genetic test for Huntington's disease. There is no trial that doesn't have that as an inclusion criteria. So that won't change for clinical trials. For observational data, for studies like the study I'm running called the HD Young Adult Study, which is studying people decades before onset because they are stage 0 and we want to do stage 0 trials, they do have to have a genetic test to be in the study. It's the same within many studies like Enroll and Image HD. Studies like Pharos didn't have to, and that they were tested and the result wasn't fed back. But, so if you want to be involved in clinical research and you want to do a trial, eventually, we don't have any trials in stage 0 or stage 1 yet. We have trials in stage 2, HDISS, which is already earlier. But if eventually when we do have trials in stage 0 or stage 1, typically people in stage 0 are going to be young. They're going to, based on what we know, they're probably going to be in their, they're going to be in their 20s, early 30s, 20s and 30s. So the whole thing about a clinical trial is it's testing whether a therapeutic will slow disease. There is the possibility when you enter a clinical trial, and we know this sadly from the Generation HD1 study, that you may get worse because we don't know what therapies will do. That's why we have to do scientific clinical trials. So to do a clinical trial, we have to be sure that that person carries the Huntington gene, because we can't, ethically, we wouldn't be allowed to do trials where a person wasn't tested and to then investigate with what's called an investigational medicinal product, which means a trial drug, which is not a kind of routine drug from the pharmacy. We're bound by that governance called Good Clinical Practice, GCP, that we have to be sure of our stages to do clinical trials, because we are testing if a drug changes the course of the disease. But we know that, we know this from generation HD1, if the dose of the drug is too high, and for lots of different reasons, the drug may make people worse. So drug trials are a clinical experiment, they're a test. And also, drug trials are also about safety. And I'll use the tominersen example again. The phase 1 trial, safe, 7 months, things it was amazing. Mutant Huntington was lowered. But then the phase 3, dose was too high, probably stage of patient was too late. I think that's a very strong part of it, because they were late HDSA stage 3. And the drug wasn't safe in that group. So that's what clinical trials are about. They are testing if it works and if they're safe. And so for that, to be in a stage 0 or 1, we have to be sure that person carries the Huntington gene, because we don't know anything about how safe the drug's going to be. So I think for clinical trials, if you want to be in a clinical trial, it will be a decision about having a genetic test. Now, that's a long answer, but it's quite a complicated area, because we don't have any stage 0 or 1 trials at the moment. So I wouldn't say rush off to get predictive testing, because there might be a stage 0 or 1 trial in 2 years' time. A reason to have a predictive test is a very personal reason. Drugs currently are being tested in HDSA stage 3 and HDSA stage 2. Now, those trials that are testing, for example, in HDSA stage 2, which is already earlier than motor onset, if those trials, and there will be many, because every industry partner is planning trials like that in the HDSA stage 2, if those trials look like they shift the dial in HDSA stage 2 and they look safe, then we will be rushing to get the regulatory approval to do stage 0 and 1 trials with that drug. But we have to do the experiment. Then there would be a potential reason that if you were a gene carrier, a family member, and you had seen the results from maybe an HDSA stage 2 trial, and you think, well, actually, I really do want to get into a research clinical trial, then that's a reason to be tested then. But it's a very, very good question. What I don't want is people to, at this stage, to rush to get tested unless they're very sure, for all the reasons you have predictive testing. But now we have this framework. There will be studies in stage 0 and 1. We need more data in patients with stage 0 and 1 to do longitudinal studies. We need more data in patients with stage 0 and 1 to be able to get the cutoffs to have endpoints. So if I wanted to start a study tomorrow in HDSA stage 0, I don't have an endpoint for that group, because there is no detectable biomarker of pathogenesis. The HD young adult study that I'm running is studying large numbers of people who are decades before onset, roughly 25 years before onset, with matched young controls, mostly in their 20s. And they're going to be really important in telling us about CSF NFL to see if we can use that. But we need more data. So at the moment, we can't do trials in stage 0 and 1 because we don't have enough observational clinical research data to be able to do that. So it's a very complex area. And I think if someone never wanted a predictive test, really doesn't want to know, then that shouldn't change. No one should ever be forced to have a predictive test. But the field is changing now, where we are moving to trying to do trials in stage 0 and 1, and for that we need more clinical research. So It's a kind of complex answer, and I'm happy to discuss this further and also get other people's views, because this is a really important area.
Yeah, Sarah, thank you so much for that. And I think from what you said, the stage 0, knowing that you have the G, the staging is important if you're in those clinical trials. It doesn't matter necessarily for the observational trials. So for people to think about that, as you gave the kind of decision tree, right? If I'm a person with Huntington's, do I want to be involved? Then I need to know. If I don't need to know. But there's still options in both categories, be it you know your status, you want to know, you can do the clinical trials. You don't want to know, you can still do observationals like Enroll. So I appreciate that.
That's right.
Complex. Yeah.
Enroll gives you the option of being involved. You have a genetic test and it's stored. So that's right. And so there is the possibility of being in observational studies where you give your samples, you do all the testing, but your genetic test is done, but it isn't shared with you. That's right. But for clinical trials, I think it's different because we just don't know if drugs, we don't know if drugs are going to be safe, and so we have to be sure who we're testing.
Yeah, that's a great point. I think a couple of questions about the other end of the staging system. To kind of summarize them, one question was, why is there not a stage 4? And to tie into that, what does it mean for people who are in later stages of Huntington's disease? If stage 3 is evidence of any change in, any evidence of change in the independence scale or total functional capacity, what does it mean for folks who are in later stages? They're later stages of the disease, and why isn't there a stage 4, including them?
So we didn't have a stage 4 because we had there was a lot of discussion about this, and we looked at the totality of the total functional capacity and the independence score. The stage 4 would be really severe HDSA stage 3. So we decided to divide stage 3 into mild, moderate, and severe based on activities of daily living. Early stage 3, some mild, maybe not doing the same job or not performing your job at the same level. You've had some changes in your job. But otherwise you're independent, doing your own shopping, cleaning. Stage 2, need a bit more help with things, need help with finances, for example, may not still be in the same job. And then stage 3 is when you need more care and more help. We are mapping the Shulson and Farn staging, 1, 2, 3, 4, and 5, onto the HDSA stage 3, and they map very well. So that will be, the Shulson and Farn staging mapped onto the HDSA stage 3 is the bit that they do map on very closely. They very closely map, but the mild, moderate, and severe, the TFC Shulson and Farn staging, the early, so the mild stage 3 would be similar to HDSA stage 3 early. The Shulson and Farn staging 2 and 3 would be similar to the moderate, and the Shulson and Farn staging would be similar to 4 and 5. So we didn't want to, I do think the Shulson and Farn still has a role in the stage 3, but we called it mild, moderate, and severe. Ongoing work is in that area, but we didn't have a stage 4. Because we didn't want to have a kind of point, we didn't have measures that, we know a change in the TFC gets you into stage 3, or a change in the independence score, but then we didn't want to cut off like a TFC below 4 or a TFC below 5 for potentially a stage 4, because it wouldn't have been the 5th percentile. So it's quite complex maths, but the maths of it, we didn't have measures that would allow us to have a stage 4, if that makes sense. It's quite a complex answer. But what I don't want people to come away from this is that we're only doing this so we can do earlier stage trials. I do think we need to do earlier stage trials if we want to stop or prevent Huntington's disease. I mean, I've been working on studies of individuals for 20 years, and I think if we want to prevent Huntington's symptoms and signs, or even prevent the brain changes, I think we want to do trials in stage 0 or stage 1 that maybe prevent any further signs or symptoms or the disease. That's our goal, which is like, can we prevent Huntington's disease occurring? Prevent Huntington's disease signs and symptoms. Lots of trials are happening in HDSA stage 3. It's a really important group. Most of the trials happening are in HDSA stage 3. Any drug that ends up being approved, like Ed and Jeff were saying yesterday, will be able to be given to everyone with Huntington's disease, and that will include stage 3 once discussions have been with the regulators. But this was done to allow us the framework to do trials across the whole stage. We already have the framework to do HDSA stage 3 trials. We already had that. We hadn't got the framework to go any earlier, which is what this is.
Sarah, thank you so much. And to everyone in the room, thank you for your fantastic questions. I know we have to wrap up this part of today's presentation, but I do want to let you know there are a ton of questions that have come in through the Q&A, and we're going to encourage you to continue putting them in. We're going to collect all of those, and we'll be doing a follow-up webinar to address some of those and others that may come up. As we've heard a couple of times already in the last day and now day and a morning, science continues to evolve. Science continues to discover new things that add to what we're learning about. What Sarah presented today is brand new to us in the community, and we'll be seeing it play out in clinical trials right now, and this will grow over time. It was said in the Ask a Scientist Anything, or Ask the Scientist Anything session yesterday, science is cumulative. As we learn more, we can do more. Thank you so much, Sarah. We really appreciate it. Thank you all so much. Keep your questions coming. We'll see you in the 10:45 sessions.